Okay. So chapter 20 and 21 are the last of the gram-negative rods. And I'm working on a little document that kind of summarizes the memory tricks that we touched on last week and the different characteristics of each group of gram-negative bacilli. So look for that in next week's material. So chapter 20, these are your miscellaneous gram-negative bacilli. It's sort of like the catch-all chapter. It's got Vibrio, Campylobacter, Helicobacter, Aeromonas. I meant to go back and erase Plesiomonas because we're not going to talk about Plesiomonas. I won't expect you to know him, but pretty much just 4 organisms. Make sure you know the colony morphology and the microscopic morphology of the organisms. The habitat in which these organisms are found. We'll talk about Vibrio has a unique environment where it likes salt water. And then Campylobacter is found in a variety of different locations. You might have heard of it as a contaminant of food, raw chicken most commonly. Discuss the appropriate specimen collection and transport and processing for maximum recovery of each organism. So we'll kind of go into depth about how to process Campylobacter because he's a little bit special. Or how to collect samples for Vibrio and Aeromonas. They're pretty straightforward. They're stool pathogens for the most part, so you'll use the standard processing and collection for stool, which is just clean container, Tupperware, yogurt container, whatever. List the media of choice for each organism. So some of these might have a special kind of media that I want you to remember and associate with a certain organism. Key biochemical reactions. We're not going to learn a whole bunch about the biochemical reactions for these guys, but we'll learn a few reactions. And then confirmatory tests. Criteria used to differentiate the different species. We'll go into that a little bit. Compare and contrast each group of organisms. And then disease states for each of these guys. So take a look at your case in point. After we talk about this chapter, we might touch on it. So Vibrio species. Has anybody heard about Vibrio before? Couple? Have you ever heard of cholera? Cholera epidemics. So this is the cause of cholera. There's 10 different species that cause human infections, and we're only going to really touch on the main one, which is Vibrio cholera. It's found in water. It's found in fresh water, salt water, and then brackish water. And brackish water is between fresh water and salt water. It's got a little bit of salt in it, but not as salty as salt water. So standing water. They saw a lot of these outbreaks after Katrina because those levees flooded and there was a lot of flooding, and a lot of humans came down with diarrhea caused by this Vibrio. So if you've got a lot of rain or a lot of flooding, your stool cultures, what you'll look for in your stool cultures might change a little bit because of those environmental conditions have changed. Run-of-the-mill, you may not screen for Vibrio unless you have a reason to. And let's see, it causes severe diarrhea, liters upon liters of diarrhea every day. And there's a picture in one of my textbooks where they have a patient laying on a cot and they've cut a hole, is not in America, cut a hole in the cot and then put a bucket under because the patient couldn't get up and down and go to the restroom. They just emptied the bucket. So primarily seen in poor sanitation, third world countries, but we see it here in America too. So Vibrio cholera is the #1 pathogen. Gastritis, cholera. It can also cause wound infections, and then if it gets in the blood, it can cause bacteremia. So majority of your Vibrio species, you'll see gastroenteritis. Okay, and cholera is a little bit different than your run-of-the-mill gastroenteritis. So clinical manifestations, they don't isolate it frequently. So it's not part of our routine stool workup. So if there's a reason to suspect it, we need to know. That way we can set up a special type of media meant to isolate Yersinia. Okay, so that's where communication from the physician and the nurses to us is very important. There are some risk factors involved, and these are the kind of things that a physician might look for when he's getting a medical history. So recent consumption of raw seafood like oysters is a big clue. Recent immigration or foreign travel. For example, maybe they came back from Haiti, and Haiti has been battling cholera for quite a while. So that's a big clue. Gastroenteritis with cholera-like or rice water stools. Rice water stools is basically a clear kind of diarrhea, but there's a lot of mucus flecks in it, so it looks like there's rice in the stool. So when you see that rice water stool, you want to think of Vibrio cholera. Okay? And then accidental trauma in a watery environment. Maybe they're fishing down in the Gulf and they cut their finger with a fish hook. That's a risk. So the general characteristics for Vibrio, let's see, big thing here is it's curved. We only typically see it from the clinical sample itself, but in your test questions, they'll probably give you that curved gram-negative rod. You can see it better on this fluorescent stain than you can in this gram stain. Maybe your book is a little clearer. So curved gram-negative rod, almost like a boomerang. It's catalase and oxidase positive. So notice I bolded the oxidase. How does that, what is Enterobacteriaceae? We talked about that group last week. What was the reaction for that group as far as oxidase? Anybody remember? Negative. Okay, so that's a good indication that it's not Enterobacteriaceae, because most of them, except for one that we didn't talk about, is oxidase negative. And then it also reduces nitrate to nitrite. Most of our Enterobacteriaceae do that. So you'll notice if you're working up a stool culture in your clinical sites, we talked about HE media and XLD media and how it made looking for stool pathogens a lot easier. Do you remember talking about that last week? What did we look for in those stool medias? They're meant to isolate stool pathogens, right? You remember which two? Shigella and Salmonella, right. So how did they look like on these HE and XLD plates? Remember? Clear colonies, right? And Salmonella had a black center because it's H2S positive. And Shigella did not have a black center because it's not H2S. So clear colonies on stool media. That's a big clue that we've got a pathogen, right? I'm just going to put SS for Salmonella and Shigella. Another thing that we do is we inoculate, that's our HE or our XLD media. Another thing that we do for stool cultures is that we inoculate a sheep blood agar. And sheep blood agar grows everything, right? So there's going to be tons of normal flora on our sheep blood agar. So what we'll do is well, here's our sheep blood agar plate, and we do a quad streak, right? And where's the heaviest amount of growth on our quad streak? First quadrant, right. So what we can do is take a swab and sweep into the first quadrant, pick up a goober of organism, right? A big old glob, okay? Sweep oxidase. And then we put the reagent on there, and we're looking for anything that's oxidase positive, or we're looking for any beta hemolysis. Okay? So that's another thing that we do to screen for stool pathogens. So if we do a sweep oxidase on our first quadrant and it's positive, we might have a Vibrio, right? So we would have to dig that guy out and do some further testing, right? All right, so here's some more general characteristics, and these are some tests that we can use to identify Vibrio. So the Vibrio stat disc is this disc that has this reagent on it. You don't have to know that. But basically, you're looking to see if the organism is susceptible or resistant to the disc. So Vibrio is, if I remember correctly, Vibrio is sensitive to the Vibrio stat disc. And Aeromonas, which is another stool pathogen, is resistant. Okay? Easy, right? There's also a positive string test. So they take this sodium deoxycholate, and that's the bile solubility reagent that we used for strep pneumo. And you just emulsify some colonies in there, and you take your loop and you touch it and you raise it up real slowly. And you want to measure the string that sticks to the loop. Real high-tech science here. Okay, so yeah. And then if it's so long, it's supposed to be Vibrio. So they don't use that much anymore. Another one is the salt tolerance test. Vibrio, a lot of your Vibrio species are salt-loving. They're halophilic, salt-loving. Know what that term means. Halophilic is salt-loving. So we take 6.5% sodium chloride and we immerse a little bit of that organism in there, and we incubate it, and we want to see if it's able to grow in that concentration of salt or not. Okay, so most of your Vibrio organisms are able to grow and withstand that high salt concentration. So your suspension will become cloudy. But Vibrio cholera cannot stand that salt concentration. It will only grow in 0% sodium chloride. Okay, so definitely know that Vibrio cholera will only grow with 0% sodium chloride. He's kind of your oddball. So here's some key features for Vibrio and Aeromonas, and I kind of highlighted the ones that I wanted you to know. See how they're all oxidase fermenters? So they kind of, if you're given a glucose, an acidic glucose reaction, and it's a glucose fermenter, you might want to think that it's an Enterobacteriaceae, right? But it's not, because it's oxidase negative. Or oxidase positive, I'm sorry. Enterobacteriaceae are all negative for oxidase, right? So the oxidase helps us differentiate it from that other big group. And here is that Vibrio stat disc. Vibrio is sensitive, and your other stool pathogen is resistant to that disc. Growth on TCBS auger. TCBS auger is specific for Vibrio. Okay, so you want to link TCBS with Vibrio. And Vibrio can grow on that kind of auger, but the other two won't. And then we have the sodium chloride test to see if it's able to grow in high concentrations of salt. And Vibrio cholera will grow only with 0% sodium chloride, whereas other Vibrio species will be able to grow in high concentrations. Notice the other two can't grow in that high salt concentration. Okay? So the disease, we talked about cholera and the rice water stools with the mucus flecks that looks like rice. A huge loss of water because there's 10 to 30 movements a day. Whole liters of water leaving your body. You dehydrate so fast from this. And that's why elderly and young children are usually don't survive from the disease. Hypovolemic shock from losing so much fluids, metabolic acidosis, and the toxin causes the diarrhea, a toxin that the organism produces, not the organism itself. And transmission is usually through tainted water or food. So specimen collection, we usually collect an aspirate if it's a wound, tissues if it's a wound, stool, run-of-the-mill stool culture. Let's see. Here's if we're going to culture for Vibrio, then we need to know that the physician is suspecting it. So we'll put it on a media specific for Vibrio. Let's see. So what's it stand for? Thiosulfate, citrate, bile salts, and sucrose. And I didn't ever memorize what that stood for, but I remember TCBS. And I thought of an ice cream cone. Have you ever heard of TCBY ice cream? They sell ice cream cone. And the gram-negative rod kind of looks like an ice cream cone. I don't know. So yellow, if it grows yellow colonies, it's Vibrio cholera. And if they're green colonies, then it's another species of cholera, or Vibrio. Okay? So definitely know that. Other species, green, Vibrio cholera, yellow. Okay? If it's not Vibrio, it shouldn't grow. Let's see. What did I say about that? I'll be able to differentiate Vibrio cholera from Vibrio, the other two types of Vibrio, or the other two main species of Vibrio, based on the sodium chloride test and growth on TCBS. Okay. Now the next organism we're going to talk about is Aeromonas. It's oxidase positive, so it's another organism that we would catch over here with our sweet oxidase on our sheep blood agar. Okay. It is glucose fermenting, just like Vibrio and the Enterobacteriaceae, but that oxidase test helps us differentiate it. It's also a gram-negative rod, but it's straight. It's not curved like Vibrio is. It can also cause gastroenteritis. That's probably the leading infection that Aeromonas causes from drinking water that's contaminated or seafood. And there's different types of diarrhea that you can see, but we're not going to learn about all the different types and how to distinguish between diarrhea. Diarrhea is diarrhea. You can also see wound infections with Aeromonas, and it's not very common, and it can really have some really disastrous effects. It can cause tissue death. There was a case of Aeromonas in the newspaper, and they had to cut some tissue out, but I can't remember the details of that story. So Aeromonas, hydrophilia is the #1 organism. Can also see septicemia and some other kinds of infections, but primarily gastroenteritis or wound infection. So here it is on sheep blood agar and McConkey. It is a lactose fermenter, and it's beta hemolytic, so that's why we also look for beta hemolysis on our sheep blood agar when we're doing a stool culture. Okay. This is kind of talking about the screening procedure for stool. You want to look for oxidase positive organisms. Once you've isolated your oxidase positive organism, you can do an indol, the string test, the Vibriostat test, and sodium chloride test. Of course, those are probably old school. When you get into your clinical site, they might have some new methodology for, they might stick it in an analyzer and be done with it. Here we go. Campylobacter. Campylobacter jejuni is the #1 isolate that we see in this. It's microaerophilic. What does that mean? If we break the word into bits and pieces, what does micro mean? Small. And then aerophilic. Loving. Yes, it can handle small concentrations of air. So we use a smaller concentration of oxygen when we incubate this organism. What about catenophilic? What's that mean? CO2. Loves CO2, right? So higher concentration of CO2. What's halophilic mean? Loves salt, right? Okay, so, and it likes increased temperatures. What temperature do we usually incubate samples at? 35 to 37. Notice here we're incubating it at 42 degrees. So Campylobacter will grow nicely in its own little environment of decreased oxygen, increased CO2, and increased temperature. And the normal flora will kind of be inhibited and not grow as well. Hallmark morphology here, definitely know this, is that seagull wing appearance. Looks like little seagulls. Here we go. Here's a good one. Almost like a spirochete, almost, but they're not. So seagull wings. Gram-negative rods. Oxidase positive, catalase positive, definitely know that. You might hear darting motility if you look at it under a wet prep, but people don't usually do that anymore. And then it's hippurate positive. Okay. So typically, majority of the times we see poorly cooked poultry spreading this particular organism. So if you see a test question with poorly cooked poultry, think in your mind Campylobacter. Okay, it's not the only thing that can be spread by poorly cooked poultry, but it's a lot of the times. We can also see it in infected pets, and birds, and contaminated water and dairy, person to person, because of the feces and not washing your hands. It's the most common diarrheal illness in the entire world. Campylobacter, #1 cause of diarrhea. What's the #1 cause of urinary tract infections? E. coli. What's the #1 cause of diarrhea? Campylobacter, yes. Very good. Not really a serious disease, just self-limiting, a couple of days. Let's see, associated with Guillain-Barré syndrome. And I highlighted the 2 pathogens on this list, Campylobacter jejuni, diarrhea, and then Helicobacter pylori can cause stomach ulcers and can lead to stomach cancer. So definitely know those 2 things about Helicobacter. It colonizes a good portion of the population in the United States, and it's able to withstand the very low pH of the stomach. It's like one of the only organisms that can actually stay in the stomach and not be killed off by that low pH. Let's see, chronic superficial gastritis is what it causes. It produces urease, so a lot of the times they'll take a snipping of the stomach and stick it in this rapid urease media and incubate it. And it should be, if it's Helicobacter pylori in the stomach, then it'll turn a bright pink color within 4 hours. And then it can also lead to ulcers and stomach cancer. So definitely know those little tidbits. Okay, so culturing Campylobacter. Not all places use all these different types of media. Most places use the Campy blood auger plate, which is a specific kind of sheep blood auger plate. I don't know what kind of additional ingredients it has compared to the regular sheep blood auger plate, but it's Campy plate. There's Campy broth, which has some nutrients and inhibitors in there that will inhibit normal gastrointestinal flora. Scuro media is another kind that you might see for Campylobacter. And then you want to grow it in a low oxygen, high carbon dioxide. So 5 to 10% oxygen, 10% carbon dioxide at 42 degrees. Okay? So a lot of places, some places they'll use the Campy plate and the Campy broth. So another thing, the Campy likes high temperatures. It also likes low temperatures. It will also grow in low temperatures. So when you first get the stool culture in, you might put some of your stool in the Campy broth and incubate it overnight in the refrigerator. Very cold temperatures. So Campy loves that cold temperatures. He'll grow, and the normal flora, Campy, will grow, and the normal flora in the GI tract will not grow. It'll just be state as it is in the numbers that it is. Okay, so Campy will grow, normal flora will not grow. Okay, then you take a little bit of the broth, like with a swab, and streak it onto your Campy blood agar plate the next day. And this is where you'll incubate in special oxygen, decreased oxygen, increased CO2, and increased temps, 42 degrees. Okay, and this is another atmosphere that Campy loves. So Campy should grow, whereas your normal flora should not grow. Okay, and then any growth that you see on your Campy blood plate the next day, you would start a workup on. First, do an oxidase. If it's oxidase positive, do any other confirmatory tests to make sure it's Campylobacter. But this way, you're weeding out all that extra crap that's in stool. Okay, all that extra normal flora that's in stool. So wherever you go, they may not do it exactly like this, but I think this kind of highlights the special properties of Campylobacter. Likes cold temperatures, likes warm temperatures. And I think this is explaining what I just did. So Campylobacter jejuni on sheep blood agar plates, I describe it as like a puddle. It looks runny looking. It's non-hemolytic, raised, round, flat. So that's a good picture here. To presumptively identify Helicobacter, we'll use Christensen's urea media. Basically just a urease tube that you would inoculate with the organism, or maybe a snipping from the stomach. You can also use the urea breath test, where you drink this liquid that has a labeled carbon in it, and then the organism degrades the urease in that drink and releases the labeled carbon, and they measure the labeled carbon. The higher amount of labeled carbon is a positive test. So just be aware of the urea breath test for Helicobacter. You don't have to explain how it works. I don't know how it works, but and then there's also some different immunological assays available, like serology and enzyme ELISA tests and immunofluorescence tests and latex agglutination. Okay, easy, right? All right, so these are non-fermenting gram-negative bacilli. Okay, and there's not a whole lot that we're going to talk about here. Describe the general characteristics of this group of organisms. What do I mean by non-fermentative? What am I talking about? What is it not fermenting? Sugars, right. So it won't ferment glucose. If we do a TSI slant on an organism, what reaction are we going to see? K over K, alkaline over alkaline. Very good. Discuss the natural habitat. We'll talk about where these guys are typically found. Describe how these guys cause infection. Recognize initial clues that make us think a non-fermenter as opposed to a fermenter. And the typical reactions and characteristic features. There's not a whole lot that we'll talk about, but we'll talk about some things. So general characteristics, like we said, these guys don't ferment sugars. So they'll either oxidize them or they'll be non-oxidizers. So if you remember way back at the beginning of the semester, we talked about these OF media, oxidative fermentative media. And we basically, you put any sugar in there, but you only want to put one sugar in there. So say all these tubes have glucose in there. And one tube will be closed and one tube will be open. And what's this on top of the closed? Oil, and what's that do? Creates an anaerobic environment. So if it's yellow on the closed tube, what does that mean? It ferments. The pH went down and it turned yellow. Just like our TSI agar, if it ferments the sugar, it turns yellow. So what if it's yellow with the open tube? What does that mean? It's able to oxidize. So this organism can ferment and it can oxidize. What about this one? Is that positive or negative? Is it fermenting the glucose or not? It is not fermenting the glucose. What about the oxidizer tube? So this is an oxidizer, right? This is a fermenter and an oxidizer. What about this one? Asaccharidic. It's not fermenting and it's not oxidizing either, right? So this kind of describes the definitions. If they're able to oxidize sugars, they'll only break down the sugars in the open tube. So that's our oxidizer here in the middle. And then if they're non-oxidizers, and they won't break down carbohydrates, and they're biologically inactive. So that's our third set there. So this is what we usually see, these two, in this group of organisms. When we do a gram stain, they can be long, thin, gram-negative bacilli or coccobacilli. They're generally oxidase positive, but keep in mind some of these that we're going to talk about can be negative as well. And then on your triple sugar iron auger, we'll see K over K reactions. Okay, so 15% of clinical infections, or 15% of gram-negative bacilli that are, what am I trying to say here? These non-fermenters make up about 15% of the clinical infections caused by gram-negative organisms. And we could see septicemia, meningitis, osteomyelitis, wound infections. And this isn't an inclusive list. We can see respiratory infections, all kinds of stuff. So majority population at risk for non-fermenting infections, or infections caused by non-fermenters, are people with poor immune systems. So your immunosuppressed people with cancer or diabetes, trauma, someone with a surgery or burns, someone who undergoes some kind of foreign body implementation, or that has infused fluids like dialysis, or these people are at risk for these kinds of infections. And we're seeing a lot more of them with a lot more immunosuppressed people in the world, right? So this is a cute little chart from your book, and I just kind of highlighted the organisms that we talk about. And these guys, a lot of these different species in this group have very unique colony morphologies. So they look very unusual on the sheep blood auger plate. And then they also have some unique odors. So we'll talk about some of those. And I highlighted them here. So Pseudomonas aeruginosa is the #1 pathogen in this group that we're going to talk about. So if you don't know anything about non-fermenters, know about Pseudomonas aeruginosa. It's a leading cause of nosocomial respiratory tract infections. It's got a unique colony morphology, a grape-like odor. Okay, I usually smell tortilla chips. I don't smell grapes, but when I'm working up a culture with Pseudomonas, I say grape-like odor, but I smell tortilla chips. And that way everybody knows what I'm talking about. Blue-green pigment or metallic green sheen, oxidase and catalase positive. It oxidizes carbohydrates, so it doesn't ferment, it oxidizes them. So you might see like a pinkish hue on the McConkey auger with some of these guys because they're oxidizers, but it's not that bright pink that you see with the lactose fermenters. It's a weird kind of light pink. So there is a rapid ID. We talked about rapid identification of E. coli last week in lab, right? So we can rapidly identify Pseudomonas without having to do all these confirmatory tests. So if you see if it's a non-lactose fermenter with a blue-green pigment and a grape-like odor and it's oxidase positive, it's Pseudomonas aeruginosa. Okay, so definitely know this colony morphology and the oxidase reaction. Clinical infections that we see, wound infections, burn patients can come down with this kind of infection. Pulmonary infections and cystic fibrosis patients, they come down with Pseudomonas quite a bit. So I want you to know for the exam, this particular patient population at risk for Pseudomonas. And we'll talk about another organism in a minute that is also a risk for cystic fibrosis patients. Is everybody kind of familiar with cystic fibrosis? It's a genetic disease. Their lungs produce a lot of mucus, and they're not able to clear out that mucus, and they are very susceptible to respiratory tract infections. So if they have an infection with Pseudomonas, it's very important that it gets resolved as soon as possible. Also see it in nosocomial infections, endocarditis, hot tub syndrome. You can see it in that. So identification characteristics, like we just said, the green sheen on the blood agar is specific for Pseudomonas aeruginosa. There's other kinds of Pseudomonas that can also produce a pigment, but we're not really going to talk about them. Most species are beta hemolytic. Not all of them, but some of them are. Citrate positive, very drug resistant. Okay? So it's very important that we find out what Pseudomonas is sensitive to and give it to the patient as soon as possible. So here's Pseudomonas on sheet blood agar. It's kind of trying to show you the metallic sheen, but it doesn't do you justice. It's almost like if you look at a fish, a scaly fish, and that rainbow-like sheen. That's what it is. That's what it is. And then here it's beta hemolytic on this side here. This is the appearance on McConkey agar, and I think this is some kind of special. You can put it on fluorescent media and look at it under a black light and see if it fluoresces. Acinetobacter species. This guy is non-modal and oxidase negative. I highlighted it because those are two tests that we use to differentiate it from Pseudomonas. Okay. It's also found in hospitals, ventilators, humidifiers, catheters. It likes moist environments. Okay. Urinary tract infections, pneumonia, endocarditis, wound infections, burns. One of the textbooks we used called it Iraqibacter because a lot of the soldiers coming back from Iraq and Afghanistan were coming back with infections with this guy. Where the organism is a very common isolate in that area of the world. The next guy is Stenotrophomonas multifida. I love saying that name. Causes pneumonia, endocarditis, bacteremia, urinary tract infections. This guy has a very unique colony morphology. It looks kind of lavender to lavender green on sheet blood agar. It's a subtle. The picture doesn't. You can almost kind of imagine a lavender color. It's subtle. But ammonia-like odor. Smells like ammonia. ID characteristics, it's oxidase negative, so that differentiates it from Pseudomonas. It's also DNase positive, which is unique, and lysine decarboxylase positive. And finally, the last one we're going to talk about is Burkholderia cepacia. It's a nosocomial infections, and this is the other organism that is can cause pneumonia in cystic fibrosis patients. This is a very serious infection, and many cystic fibrosis patients don't recover if they get this guy in their lungs. It can cause other kinds of infections. And the colony morphology is wrinkled. Notice how wrinkled it is. And it has a dirt-like odor. It's oxidase positive. It can also be very weak oxidase positive. So some people might accidentally call it oxidase negative. And it's modal. There are other species of Burkholderia. I kind of highlighted these because of, I wanted you to link the organism with the disease that it causes. So Burkholderia mallei causes glanders, which I have no idea, honestly, what glanders is, but I know that it's on the list for bioterrorism agents. And Burkholderia pseudomallei is also an organism. Myeloidosis. So just be aware that they're on the list for bioterrorism agents. They can spread very easily. They're not common here. So keep in mind. Here's a flow chart kind of illustrating the common characteristics of the 4 organisms that we talked about. So if you have a non-fermenter, the first test you want to do is an oxidase test. And if it's positive and it has a grape-like odor and a metallic sheen, it's your Pseudomonas aeruginosa. Okay? But if it's positive but it has a wrinkled colonies and a dirt-like odor, then it's Burkholderia cepacia. And I associate dirt and Burke. That helps me. If it's oxidase negative, you want to do a motility test, and that's going to help you differentiate between the 2 oxidase negative non-fermenters. So if it's motile and it's got this lavender and ammonia-like odor, then you want to call it stemol. And then if it's non-motile, it's your Acinetobacter. Okay? So it kind of helps if you memorize where they are on the flow chart, because I'll remember if Acinetobacter is all the way on the right, then I know it's negative for oxidase and motility. I know that my Pseudomonas is all the way on the left because it's oxidase positive. And then B comes before S for sure. So, hey, it works. Helps me remember. Any questions there? I've got another table that uses lysine decarboxylase, so if that kind of helps you remember how to differentiate these guys based on lysine reaction, that's going to be in the worksheet that I post in next week's material. Okay.